Hi, this video is about capillary structure. So capillaries, their main job is to conduct exchange or to allow exchange, really. And first I want to say a little about where the exchange is taking place. So right here is a capillary with some red blood cells in it. And the fluid that's in the capillary is the plasma. Now that plasma contains nutrients and wastes and oxygen and carbon dioxide, depending on where you are in the body. And those things are going to exchange with the fluid outside of the capillary, which is known as the interstitial fluid. So there will be exchange of gases, nutrients, wastes, and even water across the capillary membrane between the plasma and the interstitial fluid. There's another layer of exchange that happens after that in order to get what's needed into the cells or take out what's not needed. And that's an exchange between the interstitial fluid and the intracellular fluid, or the fluid inside the cells. So there's another set of exchanges that takes place there. But what we're going to talk about is what's happening right at the capillary, the exchange at the capillary wall. Now, there are four things that make exchange in the capillary exceptionally fast and exceptionally efficient. One is that capillaries are narrow. So the opening of the capillary, or the width of the capillary, is so narrow that red blood cells can only go through single file. This is important because that means that every cell is close to the capillary wall and the interstitial fluid. So it's easy for exchange to take place between the red blood cell and the plasma, and then the plasma and the interstitial fluid. Whereas if we had a much wider vessel and there were red blood cells in the middle of the vessel that weren't close to the capillary wall, they would have a harder time doing efficient and rapid exchange. The next thing about capillaries that makes them particularly good at exchange is that they are thin-walled. So remember that this is made up of endothelium only, and endothelium is just a fancy word for epithelium in the blood vessels. All right, so this is only a single layer, known as simple, of squamous cells. So simple squamous cells are the ones that form a flat sheet, and that's super thin, so that allows for exchange to take place quickly. Next, capillaries are actually leaky by design. So in between those simple squamous cells, there are little pores or windows that allow larger substances, the things that we may not have transporters for, to actually leave the capillary or enter the capillary. So it makes the exchange in the capillaries even faster. It doesn't necessarily have to go through one of the simple squamous cells. It can go around them. The last thing is that the velocity or the speed of blood flow is the slowest in capillaries. Right? So this is sort of counterintuitive because we talked about pressure and how pressure was highest in the arteries and then got lower in the capillaries and was the lowest in the veins. And our minds would like to trick us and say, okay, well then obviously that means that the blood is moving fastest in the arteries, a little slower in the capillaries, and a lot slower in the veins, but that's not the case. Even though the pressure is not the lowest in the capillaries, the velocity or the speed of blood flow is. And this is because of something called combined cross-sectional area. Okay, so let's take a look at what that means, the combined cross-sectional area. Now let's make a quick graph here. And then down here are going to be the vessels, and over here will be the combined cross-sectional area, which I didn't leave myself enough room to write all the way out, so we're just abbreviating it CCSA. Now, if we look at the various vessels in the body, let's say we have the great arteries, which I'm just going to say GA, and then we have some of the smaller arteries, and then we have arterioles, and then we have capillaries, then we have small veins, I'm ignoring venules, and then we have big veins. So first, let's just take a look at the cross-sectional area. All right, when we say the great arteries, let's just do the systemic side of the system. The great artery is the aorta. So if I were to cut the aorta, do a transverse section of the aorta to get the cross-section or to see the diameter of the vessel, let's say that it would occupy about that much of the cross-sectional area. It's just one vessel, so all I have to do is measure just that one vessel. But that one vessel splits and becomes smaller arteries. And so even though those smaller arteries, each one of them has a smaller cross-sectional area than the aorta, if we combine all of them together, there is more cross-sectional area. Same thing with the arterioles. They're much smaller than the small arteries, but there are many, many more of them. And so the total combined cross-sectional area for arterioles, all right, so I'm adding in all the arterioles that we've cut here. The combined cross-sectional area for arterioles is even higher. Now, those arterioles branch and become capillaries. And capillaries are so tiny that I'm not even going to make circles, I'm just going to make dots. But there are so many of them that if I combine them all together, the combined cross-sectional area of the capillaries is significantly higher than the combined cross-sectional area of the arteries. And the same thing is true with the small veins and veins. The capillaries come back together, form venules and small veins, and if I add those together, all the cross-sectional area of our veins, there is less cross-sectional area than what we saw with the capillaries. And finally with the vena cava, of which there are two, if I put both of those in there, there's even less cross-sectional area. So the cross-sectional area, or the total space that the blood has to go through in any particular vessel, is greatest in the capillaries because there are so many more of them than there are the other vessel types. Now, what this means is that the velocity or the speed of the blood flow is the opposite. If, let's say for each one of these, I'm going to pump through 5 liters per minute. If I have to get 5 liters of blood through this tube in 1 minute, then it's got to go pretty fast. But over here, I've got more space for the blood to spread out, and so I can get 5 liters of blood through much slower. So the greater the cross-sectional area, the slower the flow. So the flow is fastest here and here because the vessel or the combined cross-sectional area is lower. And it is slowest here where we have the greatest cross-sectional area. So, and I don't know if you've ever seen this, but if you look at a reservoir, so you have this big body of water, and usually at one end there's a dam. Now, if you go, let's say 50 feet back from the dam and you sit there in a canoe, it's not going to feel like the water is moving at all. It's very slow. And that's because all of the water is moving at the same rate and it's spread out through the entire reservoir. But by the time you get to the dam, which is a much smaller area that the water is allowed to spill over, the water is going really fast, and that's because it's got less area to get through. And yet it's moving the same volume of water as is moving back here. It's just that that volume of water has to get through a narrower space and therefore moves faster. So this is the slowest because there are the most capillaries, or the most vessels in terms of vessel types, for the blood to spread out in.